The overall goal of this procedure is to load nanospheres onto microtubules which are gliding on a surface coated with kinesin motors. This is accomplished by first coating the surface of a glass flow cell with Kinesin solution. The second step of the procedure is to adsorb kinesin motor proteins onto the flow cell surface. The third step of the procedure is to add the microtubules. The final step of the procedure is to add the streptavidin molecules which will bind to the microtubules and then add the nanospheres. The nanospheres bind to the flow cell surface where they may get picked up by the microtubules. Ultimately, one can observe how nanospheres are picked up by microtubules and are transported across the surface using fluorescence microscopy.
Hi, I'm Yolaine Jeune-Smith from the laboratory of Dr. Henry Hess in the Department of Materials Science and Engineering at the University of Florida. Today, we will show you a procedure for the inverted motility assay. We use this procedure in our laboratory to study loading of cargo onto microtubules and movement of molecular shuttles. So let's get started.
Prior to the start of this procedure, freshly prepare all stock solutions as described in the written portion of this video. Once stock solutions have been made, begin microtubule preparation by pipetting 25 microliters of growth solution into a 0.65 milliliter microcentrifuge tube. Then, add 6.25 microliters of the growth solution to 20 microliters